not exist because all the enzymes which are working in your body, all that hemoglobin which is carrying the oxygen, which is carrying the gases, nutrients, all of them are made from the proteins. All the membrane proteins we have learned together, which take part in the active diffusion and in the facilitated diffusion, all of them are made of proteins. Okay, that's why it's very important for us to learn the proteins. This is super must know thing. Okay, the proteins, all that membrane proteins that work, all the hemoglobin that's working, all the enzymes in our body, all of them are made of proteins. So proteins is a bigger structure. For example, this is the phone, right? So this phone is a big structure. What it is composed of, it's composed of many tiny what?
Many tiny amino acids.
Yes, many tiny amino acids. So inside we have the amino acids. How many amino acids we have totally? The types of the amino acids.
I remember more than 20.
It's not more than, it is 20, and that's it. 20 different types of amino acids we have, and all of them share the same backbone. What's the backbone of amino acid? If we look at the chemical structure, what is unchangeable? What's inside?
As I remember, carbon is the main thing that like is everywhere in every part of a body, DNA, RNA, carbon.
Yes. Okay. Yes. Carbon. We have the central carbon, right? We have the central carbon. And what else?
Something P, as I remember, the letter P.
Not phosphorus, actually. So we have the central carbon. We have the carboxyl group. We have the carboxyl group in amino acid, right? What's carboxyl? How it's written?
Carboxyl. Hmm, I missed this. Let me.
COOH, right?
Oh,
COOH.
this is
This,
carboxyl.
yes,
COOH.
this is the carboxyl group. You should know it, okay? So this is the carboxyl group. So what else do we have in amino acid?
In amino acids, we have monomers of proteins.
No, amino acid itself is a monomer. What's inside? I said we have the central carbon, we have the carboxyl group, and what else we have?
We I think I disconnected. Amino group, carboxyl group, and R group.
Yes, and we have the R group. What makes 20 amino acids different? Which group
The
that you
R
have
group
come?
makes all of them
Yes, R group.
different.
Okay, what's the simplest R group? What's the simplest amino acid?
The simplest one, I think, it's a H.
H hydrogen, that the one that's made with hydrogen, right? In the R group we have the hydrogen. It's H. Yes, it's H. So this is the hydrogen. Which amino acid is that?
It's uh, forget how it's
It
been
is.
called.
It is glycine. It is glycine. Okay. Glycine. Glycine. We say it glycine amino acid.
Uh,
Okay. So good. What about cysteine? Cysteine, R
cysteine.
group of the cysteine consists of what?
Oh, cysteine. I think cysteine. Write it. It's a bit bigger, yes. It's like
It's
C.
big. It contains the carbon and sulfur with S, with big S. There is sulfur and carbon,
Oh
C,
yeah, yeah.
okay? There is carbon and the sulfur, and good. So my next question is, what's the difference between the atom and the molecule?
Atom and molecule. Atom is the smallest, is the smallest thing in the world, and the molecule is a bit, just a bit bigger,
Mm-hmm.
and the atom is how to say, is the thing that is you cannot just separate it. Is the small, is the smallest thing. It can be smaller, but molecule can be smaller version of a molecule is an atom.
Yes, and two atoms combine together to form one molecule, okay? So for example, the chlorine, in the bottom of the chlorine, there is a two. So two means molecule. Two chlorine atoms join together to form a molecule, okay? What's compound?
Compound is as I remember, was it compound? Oh, yeah, okay, okay. So compound is it's like two, let's say H two plus oxygen, it's H2O. So that's compound.
Yes, compound when two different elements join together. For example, carbon tetrachloride, right? So we have the carbon and chlorine, so they are different. So that's why we say it as a compound, right? That's good. Mahbuba, can you hear now? You have to unmute yourself. Mahbuba. Oh, she has a problem with, I guess, with the
Connection.
network. Okay, what's organic? What's inorganic, Malik?
So organic is the molecule that has carbon, that includes carbon inside it, and inorganic that it's not having any carbon inside it. But I remember there's exception. It's CO2, because I remember.
Yes, CO2, carbon dioxide. It contains the carbon, but it is not organic molecule, it's inorganic molecule. Yes, that's right. So, and we have the chemical reactions. In the chemical reactions, we have things that are on the left side and things which are on the right side, right? Products are located on which side and reactants located in which side?
Oh, so products, as I remember, is on the left side, if I'm
Products
not
always
mistaken.
on the
Let
products
me double.
always on the
Oh, products always found on the right side. Yeah.
yes, products always on the right and
Rectangles always on the left.
reactants always on the left side. Okay, good. So if you combine the hydrogen with oxygen, what's going to get? What you will get?
Hydrogen with oxygen,
Yes.
H2O,
H2O, what is it? H2O, what is that?
water.
It's water. Yes, you will get the water. Okay. What's the simplest element in our body?
Simplest one, I think. Like carbon is the simplest one. I think carbohydrates.
We have the simpler one, which we call
Simpler.
hydrogen. Hydrogen is the simplest. Okay, it has one
Oh,
proton, it has one electron, but the carbon is a complex structure. It is a bigger structure, but we have special one, which is hydrogen, simplest element. It contains one proton and one electron. Okay, and forms water when it combines with oxygen. Okay, good. So what about nitrogen? So what is nitrogen and where it's contained?
Nitrogen. Oh, it's a key compound of proteins.
Yes, it is inside of the proteins. It's inside of the proteins as a amino group. As an amino group, it's located inside of the proteins, right? So in the amino group we have the nitrogen. Okay, phosphorus. Where can we see the phosphorus?
Phosphorus, nucleus acid, and a nucleic acid and a DNA RNA structure.
So nucleic acid actually mean the DNA or RNA. So we can see the phosphorus inside of the DNA and RNA. Where else can we see?
This ATP, the I forgot how it's
Inside
uh
adenosine,
adenosine is
adenosine triphosphate. Yes,
adenosine
adenosine triphosphate.
triphosphate.
What is ATP actually?
I think it's a source of energy.
Yes, it's the source of energy in our body. So what else? Where can we see the phosphorus?
Mm, think I thought that that's the only places that you can see. Need to check.
We can also see them in,
In the back of a DNA.
no, actually you said the DNA in the
Yeah, I
backbone.
did say
So you said the backbone of the DNA and RNA. You said the ATP. Where else can we see the phosphorus?
cell membrane structure.
Yes, of course. In the cell membrane, we have the phospholipids, right? We have the phospholipids. So the phospholipids, we are saying phosphor. There is a phosphate, okay? There is a phosphate inside. There is phosphorus inside. So that's why we say it is also in the cell membrane. So, okay. So what's the difference between organic and inorganic molecules? You said about it, right? So you know the table salt, the salt which we use, is it organic or inorganic?
It's unorganic.
It's inorganic.
That's all.
Hydrochloric acid,
Unorganic.
okay, DNA,
Organic.
methane,
Organic.
ethanol,
Organic.
table sugar,
Also organic.
also organic. Good job. So biomolecules, four types of the biomolecules. Can you count them?
Biomolecules. This is the I think the most confusing part.
Four types of the biomolecules.
Oh, actually, yeah. Okay. So, nucleus acids, lipids, carbohydrates, and proteins.
So nucleic acids, lipids, carbohydrates, and proteins, they are all biomolecules. They big molecules, okay? We call them biomolecules. Okay. So biomolecules, the chemical compounds, they all found in the living organisms, okay? For the vital, they are very vital for their survival, for their life, okay? They're mainly composed of what? The biomolecules are composed of? They composed of carbon, they composed
Carbohydrate.
of oxygen, and there is nitrogen inside, phosphorus inside, and sulfur inside. And all of them that the biomolecules are going to contain. So we said about the four major types. The first is, as you said, they are carbohydrates. Carbohydrates are main sources of the energy in our body and structural support. For example, glucose, you heard about glucose. This is also carbohydrate, a type of the biomolecule. Or sucrose, saccharose. The saccharose, this is the table sugar that you eat on a daily basis, maybe not. And also the starch, the krahmal, and cellulose. This is also the cell wall of the plants, right? And all of them, the carbohydrates, they are the type of the biomolecule. And we talked about in the last lecture, we talked about the proteins. Proteins, they build and repair our tissues. They make the enzymes. They make the hormones. Even all of your hormones are your proteins. They are protein based. Okay, that's why it's important. And also, they support your immune system. They play a key role in your immune system as well. That's why it's important to know the proteins. Okay, so proteins, they made of amino acids. They are building blocks, right? The monomers of proteins are amino acids. The example to the protein, as I said before, hemoglobin. It can be any hormone. For example, the insulin. You heard about insulin, right? And
Yeah.
also the other enzymes. All the enzymes in your body are protein based. And about the lipids, you know what are the lipids, right?
Lipids, I did write down. You asked from a last lesson. You also asked it.
So
Wrote it
lipids,
down.
жиры. На русском переводится как жиры, хорошо?
Mm-hmm.
So, функция липидов — это store the energy. They store the energy. They form the cell membranes. For example, phospholipids, they are also lipids that make up the cell membrane, right? And also the examples for the lipids are fats, oils, phospholipids, cholesterol, steroid hormones, all of them are lipid based, okay? So nucleic acids, they store and transmit the genetic information. So they store and transmit the genetic information. This is the function of the nucleic acid. If someone asks you what's genetic information, you say it is DNA and RNA, right? So DNA and RNA, this is a part of the nucleic acids. And also all the nucleic acids, they made of the monomers, which we call the nucleotides. Okay, they have the nucleotides. Example for the nucleic acid is DNA and RNA. Okay, this is just general explanation. So we stopped in the proteins, right? What's dehydration synthesis reaction, Jahangir?
Dehydration synthesis reactions. I think from a name, it's pretty obvious that it's dehydrating the place. Like, I don't know how to explain, but the water is going out of the amino acids. The water is just leaving amino acids.
Yes, exactly, you're right. When this happening, when forming the bond or breaking down the bond, in which case?
I think it's more in a making bond because when they
Yes.
follow up to each other, water needs to go out.
Of course, when they make up the bond, when they want to make the bond, when they want to join two amino acids together, they form which kind of bond? What's the name of that bond?
The fourth bond,
No,
right? Sorry,
peptide bond.
a peptide.
Peptide. It's peptide bond, okay? So it is a peptide bond. So when you want to make when you want to make two amino acids together, between them, you should have a bond. Okay, and this bond is called peptide bond only in case of proteins. Okay, we call them peptide bonds. Okay, good. And this process is called, when we form the bonds, dehydration synthesis reaction, right? So what is then what's hydrolysis reaction?
Hydrolysis reaction, I think it's totally opposite of a dehydration
Synthesis reaction?
reaction.
Yes.
I think it's
So
like take I don't know how, but it's like not pushing water inside it, just letting the water stay inside.
it's like it's like breaking down the bond, breaking down the peptide bond with the energy of water, with using the water. Okay,
Yeah.
with using the electrons of the water, we're going to we're going to break down the bonds, peptide bonds. So while breaking down the peptide bonds, we use the water. When we use the water, this is called the hydrolysis reaction. When we release the water, it's called dehydration synthesis reaction, right? So how many peptide bonds in 10 amino acids? I joined the 10 amino acids together. How many peptide bonds will be there?
I think 9.
9 peptide bonds. Okay. While joining 10 amino acids together with 9 peptide bonds, how many water molecules were used?
I think none because they're already inside it.
Yes, we release the water. We don't use the water. We release the water. How many water molecules were released while making 9 peptide bonds?
9.
Of course, 9. You see, in the last lecture, you were just sitting, not understanding. Now you understand. It's good. Okay, that's I want from my students to know. And these are the key concepts. That's good. You know the dehydration synthesis, you know the hydrolysis reaction now. That's really good. Thanks for your actively learning. So in today's lecture, we're going to start the protein structure levels. We are more doing focusing on the levels, okay, organizations of the proteins. Let me join my iPad. So online classwork. So, so can you see my screen now?
Yeah.
Yes, thank you for letting me know. Good. So protein structure levels. So let's talk about how the proteins are structured, how they are organized, okay? So what's going on here? So what's happening in here? In here, this is the protein structure level. This here is illustrated. And what's happening actually? Once the amino acids form the polypeptide chain, right? As you can see here, we have the polypeptide chain here, right? This is called polypeptide chain or polypeptide bonds. This is one amino acid, this is another, this is third, this is the fourth amino acid. What's linking them? What is it?
It's a bond, the
What?
peptide bond.
What? Yes, it is peptide bond. Peptide bonds are exist only in the proteins. Okay, while joining the amino acids together, right? So this polypeptide chain, always remember, is not functional protein yet. Okay, it's not the functional protein that we want. It needs to fold and it needs to twist into the correct shape to work properly in your body. Okay, so there are 4 levels of the protein structure. The first is primary structure, the secondary structure, the tertiary structure, and we have the quaternary structure of the proteins. Okay, so what's primary structure? The bad of the necklace. So everyone has a necklace, or everyone saw the necklace once in their life, right? This is just the linear chain. This is the linear chain of the amino acids. They are held together by the peptide bonds. These yellow ones are the peptide bonds. They held together by peptide bonds. Okay, why it matters? Why it's so important? Like the letters in the word, the exact order of the amino acids determines everything that follows in this case. Okay, so this is the primary structure. The second contains bonds are joined. Peptide bonds join the amino acids together. So secondary structure, the chain starts to fold into local shapes here in the secondary structure. Okay, so this kind of linear chain is going to fold. They're going to change its size, its shape. Okay, so for example, alpha helix. This is the shape also, like a spiral shape. Okay, like a curly hair. And we have the beta plated sheet. This is the zigzag shape, okay? And these shapes are held together by, now we call them hydrogen bonds, okay? They are called the hydrogen bonds. So does it matter? Imagine I have the first amino acid, second, third, and fourth amino acids, right? What I want to do, I want to make more interactions between the amino acid. First amino acid is not interacted with fourth one, but it will be possible if I make in this shape, if I make in the curly shape, if I make the spiral shape like a curly hair, I can do this. I can do this folding, right? I can, in case of the zigzag shaping, in case of the coiling like this, I can make more interactions between the amino acids. I can interact the third with the fifths or first with the sixths, right? There will be more interactions with making the hydrogen bonds together, okay? These are the hydrogen bonds as well. This is just making the zigzagging pattern, and this one is coiling pattern, okay? So we have two patterns. First is alpha helix. This is the coiled structure, and beta plated sheet. This is the zigzag pattern. Okay. So why we need this folding? Why to make a complex life? Why should I do this actually? Why our body does this? So these folds add extra strength and structure. Okay. Good. So this gives us more strengths. They will be more stable. Here, the amino acids are not stable. They can be disturbed or they can be broken down very easily in case of the primary structure. But in case of the secondary structures, they held by extra bonds, which we call the hydrogen bonds. Okay, that's why it's more important. If I ask you primary actually structure of the proteins, you have to say they just simple amino acids connected with peptide bond. If I ask you secondary, you should say there are two types of the secondary structure, beta sheet and alpha helix. Alpha helix, they are coiled in structure, beta helix zigzagged in structure, okay? Good, any questions?
Now getting all of this.
Okay, and we have the tertiary structure as well. So what's tertiary structure actually? Now the whole polypeptide falls into the compact 3D structure. Okay, so they're going to make 3D. This is the beta sheet. This is the alpha helix. They're going to join together with extra bonds. So we know that here we have the hydrogen bonds, here we have the hydrogen bonds, right? So, and after that, they're going to combine with disulfide bonds, okay? So the disulfide bonds. They're going to join with disulfide, or we can say disulfide bridges. They are strong bonds between cysteine residues. I'm not going to stop in the cysteine as far as it is like cysteine is an amino acid. There will be an extra amino acid. We have 20 types of amino acids, and cysteine is one of them. They're going to do the two what? Two disulfide bridges. This is one sulfide, this is second sulfide. They're going to join together to form a stronger bond. bonds here. So this is extra bond, okay? And this is called the tertiary. Then alpha and beta, alpha helix and beta sheet are going to combine with disulfide bonds, and they have extra hydrogen bonds, right? They're going to form this 3D shape. 3D shape is the tertiary shape, okay? So this 3D shape decides the protein's function. Okay, so what I am saying, this 3D shape is going to decide the protein's function. It's like a key that fits into the lock, which we call the enzyme. And this shape, 3D shape, is going to what? It's going to fit to the active site of an enzyme. Imagine this is the active site, and this is the enzyme, right? So this one where it should fit actually. Okay, 3D shape decides the protein's function like a key fitting into the lock. Okay, this is a tertiary shape. And what's quaternary shape? Actually, quaternary, this is like teamwork. It's like a teamwork. Some proteins are made of more than one polypeptide chain. We know them. These chains join together into one working protein substitutes, okay, two alpha to beta chains. So, what I mean by quaternary structure, a lot of tertiary structures are going to combine to make one quaternary structure, okay? So in this diagram, it's illustrated already, right? So as you can see, this is the primary structure of the protein. This is the secondary structure of the protein with hydrogen bonds. What a lot of amino acids are joined together with hydrogen bonds. It's more stable. It's coiled. This is alpha helix, and this is alpha helix and beta sheet combined together to form one tertiary structure. And a lot of tertiary structures, like in this hemoglobin, I'll have four chains here. I have four subunits here, two alpha subunits, two beta subunits here. All right. So they are making the quaternary structure. Quaternary when the tertiary structures join together. Okay. So why this classification, why this structuring is important? Here you can see the primary structure with amino acids. We have 20 amino acids. All of them are here. And this is the alpha helix beta sheet. They folded into it. This is the tertiary structure. This is the quaternary structure. For example, this is hemoglobin. It's also made from the proteins. So why this organization so important? What's the significance of it? So each level adds structure, each level adds the stability, and in each structure we have a unique function. If one part misfolds, for example, if a problem in misfold, if problem here in folding the primary structure, or problem in folding the secondary structure, problem in the tertiary structure, if any problem, what's going to happen? The protein might not work properly. And this causes a lot of diseases. For example, sickle cell anemia or Alzheimer's disease. All of them can happen. Okay, that's why, and this is very important. Okay, any questions? This folding is really important.
No questions, just processing all this information.
Okay, got it? Got the general idea?
Yeah,
Why
generally,
we're learning it?
I get the idea.
Okay, good. What about others? Others, any questions to me? محبوبة قول خيو. Any questions? Did you understand? Jahangir,
Yeah.
uh, this is the first time you're joining, right? Oh, sorry,
No.
not Jahangir. Sorry. We have another. Nur Hayat. Oh, she's not here. Sorry. Gul Hayo, Mahbuba, you here? Okay. Kulkhaya always is entering and Kulkhaya. Okay, good. So, is it understandable? Can you understand the process? Is it understandable? Yes, yes. Okay. So quaternary structure. What's hemoglobin actually? What's quaternary structure? Let's discuss. So, quaternary structures, I said, multiple polypeptide chains. For example, a lot of tertiary structures are going to join together to form the functional protein, right? For example, the hemoglobin made of multiple subunits, right? And the fourth final, this is the fourth final level of protein structure. It only exists in the proteins made of more than one polypeptide chain. If in the protein we have more than one polypeptide chain, so this is the quaternary structure. For example, hemoglobin has four subunits, two alpha helix, two beta sheets, right? So here, alpha, two alpha helix, this is alpha helix, this alpha helix. Here we have beta sheet and we have the beta sheets. Okay, two alpha and two beta chains working together to carry so correct folding is always important. If the protein folds in the right way, it does its job. For example, enzymes, transporters, antibodies are all of them going to work. Wrong folding, useless or harmful proteins can happen. For example, a tiny mistake in amino acid folding, what's going to happen? Hemoglobin folds incorrectly. Okay, this can cause the sickle cell disease. Any tiny mistake in amino acid folding or orders even, if you have an order problem here, if the sixes mixes with the third one, if incomplete, if wrong order, wrong shape, can happen. Wrong shape can't work properly, and that can cause the disease. Okay, so in the sickle cell anemia, one wrong amino acid leads to hemoglobin folding incorrectly, or Alzheimer's disease. This is the protein folding problem as well in the Alzheimer's disease. Misfolded proteins form the plaques in the brain. So wrong proteins are will be build up in your brain. So in this case, it's a big problem. So structure equal to the function always, okay? Structure, correct structure, correct function, okay? Primary structure, amino acids in a straight line, right? So after the secondary structure, they make the coils, right? Or they're gonna sheet in a tertiary structure. The whole chain fold in understood? Can you hear me?
Yeah, I can hear you, but sometimes you break down.
Oh, I'm really sorry. Now it's clear, right?
Yeah, now it's
Can you
clear.
see me now?
Yeah, I can see.
Okay, thank you. You can see me?
Yeah, I can.
Okay, good. Thank you for letting me know. So let's do it. So first, who's going to start? As always, Jahangir is going to start. Jahangir, you can read the first one. Can you see my iPad as well?
I see only the structure, amino acid, peptide bond, different structures.
But now, can you see the screen?
Now there is nothing on the screen, just empty.
Oh, just a moment. Oh, you can see it, right?
Yeah, now I can see.
Okay, Jahangir, can you do the first one, please?
The bond that holds two amino acids together is called I just answered it a few minutes ago.
The bond that holds two amino acids together is called what kind of
Peptide bond?
bond? Of course, it is peptide bond. Yes, peptide. Okay, the second محبوبة. محبوبة كفروة. You can start the second one. Okay, Gulhaya Akbarova. Okay, removal. She said removal. Okay, and this process is called what? محبوبة. Dehydration. Yes, she's right. Removal and dehydration. Reaction synthesis reaction. Action. Okay, Gulhaya, your turn. The third one. Monomer is a monomer of the protein. That's right. The fourth one, Jahangir.
Because proteins are essentially chains of amino acids linked together by peptide bonds. A protein might also be called polypeptide.
Of course. You're good. Fifth one, Mahbuba. Hydrolysis, yes. Hydrolysis. And involves addition of water. You're good. Nice. Good job. Any questions to me?
I get no questions.
Any question? Okay, let's start the carbohydrates. What are the carbohydrates? Carbohydrates are quick energy resources for our body, okay? They give us a very quick energy. And carbohydrates, and this is the another big branch of the biology. We learned the proteins, it's a big, right? And the carbohydrate is also big. So carbohydrate, they have their own bonds, they have their own monosaccharides, okay? So they have their own monomers even. So monomer of the carbohydrate is called saccharide. Can you see my screen?
It's still on questions.
Just a moment. What about now?
Still questions.
Okay, we're turning into the carbs. You can see my screen right now.
Yeah, now we can.
Okay, good. So in the carbohydrates, they have their own monomers, and the monomers are called in this case what? The monomers here, we call them saccharides. Okay, the saccharides are monomers. Okay. What's important here? What you should know regarding the carbohydrates? It gives us very quick energy, and for example, all the cookies that you eat, right? All the cookies, they contain the carbohydrates. All the bread that you consume also contain the carbohydrates, okay? So is it audible? Can you listen me?
Yeah, we can hear you.
You listen me, right?
Yes, yes, right.
Okay, okay, good. So the unique feature of the carbohydrate is that the monomer of the carbohydrate is called what? Saccharide. The monomer itself is called what? Saccharide. Okay. So one sugar unit means one saccharide unit carbohydrate. So carbohydrate actually it's the big units inside all the saccharides. One sugar unit, one saccharide. Many sugar units become carbohydrates. Okay. They made of what they made of? They made of carbon, they made of hydrogen, they made of oxygen. Okay, they made of carbon, hydrogen, and oxygen. Their ratio, carbohydrates one. Okay, in case of one carbohydrate, you have two hydrogen you will have, and one oxygen you will have. Okay, in case of glucose, you have six carbon, 12 hydrogen, and six oxygen. Okay, this is for the glucose as an example. What makes carbohydrates very special actually? So carbohydrates, they are actually carbohydrates, the only macromolecule where the single monomer, one unit is called the carbohydrate, okay? You even call the only macromolecule here, the unique feature is written. This is the only macromolecule where a single monomer, for example, the saccharide, is considered as a carbohydrate. Can also be considered as a carbohydrate. Okay, so proteins and the nucleic acids, they need the long chains to be functional, but one saccharide is enough to count as a carbohydrate. Okay, in the protein you need the long to be functional. In the nucleic acid, the saccharide case of carbohydrates, one saccharide is enough to count. Okay, saccharides. So without the monosaccharides, we can't start the carbohydrates. We have types of the carbohydrates. They are called monosaccharides, saccharides. We have disaccharides and, and polysaccharides. We have types of the saccharides. Okay, mono, D, and polysaccharides. Monosaccharides mean sugar immediately. was my nigger. Here you can see only one fructose. Can you hear me?
Yeah, we can, but sometimes you just disconnecting. I think.
I'm so sorry. So monosaccharides, the second is first is glucose. You can see here this is one unit saccharide. This is the another one unit saccharide. This is another one unit saccharide. Okay. So glucose, fructose, or galactose, all of them are monosaccharides. Don't ever forget, okay? So glucose, fructose, galactose, they are all simple sugars. They all called monosaccharide. For example, glucose is a blood sugar. Okay, they are used by every cell in your body for the energy purposes. The glucose, okay, this is the blood sugar. Or fructose. Fructose, this is found in fruits. Galactose. Galactose, they found in its own. Okay, so galactose is found in the meal, fructose in the fruit, glucose is found in blood sugar, blood. Okay, the chemical formula of the monosaccharides stays the same. The formula for the fructose, galactose for the glucose here is not. Okay. So here OH regarding this. Can you hear me?
Yes.
Okay, good. So carbohydrates, source of the energy. For example, we have the simple sugars. For example, the soda, they give us very fast energy. We have also the complex starches. We call the starches are krahmal. Okay, for example, in the pasta, in the potatoes, we have the sustained energy. There are energies. And regarding the function as well, carbohydrates, they are immediate energy sources or stored for the later as a glycogen or fat. And metabolism of the carbohydrates, they quickly down for the energy purposes. For example, if you eat carbohydrates, for the dinner or for the breakfast as well, they're going to break down very fast. You will lose them very fast. They quickly broken down for the energy purposes. Okay, they're not as stable as the proteins. And excess stored as a glycogen. You're eating the glucose. If you eat them much, if you eat the glucose very much, what's going to happen? They're going to be stored as a glycogen in your body, in your muscles, in your liver. The glucose will be stored as a glycogen. It will be stored as a glycogen for the later usage. Okay, for the later usage when you will be hungry, or when you're fasting, this glycogen will be broken down into the glucose again for the energy purposes. Okay, or excess one will be broken down into the fats as well. It's not only for athletes to take a lot of quick energy, it's essential for the brain. For example, when you're working, you only want to eat sweets or chocolate, right? Am I right? So this is essential for the quick energy. You're using the energy, your brain wants energy, you eat them. And for the muscles as well, they are essential for your muscles, and it's essential for the daily function as well. So after that, monosaccharides, glucose and both the fructose, they are all monosaccharides, but main difference, the oxygen in the glucose located in the first carbon and oxygen in the fructose is located in the second carbon. This is just the linear structure of these molecules. Um, Glucose can form the ring structure as well. There is a ring structure as well. You can see the ring structure here. And also disaccharides are two monosaccharides are going to combine to make the disaccharides in this case. For example, glucose and fructose, two monosaccharides, are going to combine together to form the sucrose molecule. Sucrose, this is the disaccharide, which is a table sugar, saccharose, table sugar that you eat every day. This is the disaccharide combination of the glucose and fructose molecule. But before binding them together, you have to make the glycosidic bond, not peptide bond like proteins. They make the glycosidic bond, special bond in the carbohydrates. Okay, so glucose and fructose, they're going to form together. They're going to join together to form the sucrose. This is the table sugar. Okay, any questions, guys?
For now, no questions. Still processing
So
all this information.
okay, glucose and fructose, they're going to join together to form the sucrose, okay, saccharose. So two glucose molecules are going to join together. Two glucose molecules are going to join together to form the maltose, okay, to form the maltose molecule. So sucrose, this is the combination of the glucose plus fructose. Maltose is a combination of two glucose molecules. Okay, here you can see maltose combination of two glucose molecules. Okay, uh, let's say the formula of the glucose is this. The formula of the fructose the same. What will be the formula? What will be the formula of sucrose? What will be the formula of sucrose if I join them together?
C12 H24 O12.
So you're saying C12, H24, and all 12, right? Who thinks so? Please raise your hands. Any other ideas? Munisa Temirova, why you think so? Why? Please explain us.
I heard it. It's cool.
Huh?
I've heard this at school, but I don't know why.
Okay. Um, this should be like this, right? If I combine together the glucose and fructose and I'm getting the disaccharide, so what's going to happen? The formula must be like this, but not. Why? Because this is the dehydration synthesis reaction. This is dehydration synthesis reaction. We are removing the water by joining these bonds. I'm making a bond. I'm releasing the water from this molecule. I'm releasing the water. Okay, what's going to happen? The dehydration synthesis is happening. Hydrogen and OH is going to release one, water will be released. And when you look to the formula, if you subscribe to one uh, water molecule, so what formula you will get? C12H 22 and O11. Okay, so you remove one water to make the disaccharide. That's why the formula changes for the disaccharides. Okay, so this is important. This is not like this. Okay, it's wrong. You should release one water, and at the end you will have this formula, right? Okay. Any questions? Everyone got that point?
No questions.
Okay, so glycosidic bond, polysaccharides. You learned this bond. If the bond in the proteins we call the peptide bonds, right? But the bond in case of the carbohydrates we call them glycosidic bond. Okay, don't confuse between them. Is my screen visible?
There is visible.
So polysaccharides. Can you see the polysaccharides in your screen?
I see glycosid. Now I see polysaccharides.
Okay, so polysaccharides. Examples for the polysaccharides. Polysaccharides means a lot of monosaccharides are joined together. So a lot of monosaccharides, they join it together to form the polysaccharides. For example, the cellulose, the starch, the glycogen, the ketene. What was the ketene? Who knows? What's ketene?
The cell wall of fungi.
The ketene is the cell wall of the fungi, yes. The glycogen, what's glycogen? Glycogen is blood sugar actually in our body. So glycogen is stored. A lot of glucose molecules are joined together. Not two. If two glucose are joined together, we call them maltose. But here, a lot of glucose molecules joined together and stored in our body. This is called the glycogen. Okay, and starch. The starch and the glycogen, they are very similar in their structure, right? They are very similar in their structure because the starch, this is a plant glucose storage, and glycogen, this is animal glucose storage. This is highly branched for the quick energy release, energy reservoir in the roots and in the seeds. This is in the starch. For example, starch means krahmal, glycogen means glycogen. So starch is a combination of a lot of glucose chains. This is the polysaccharide, which is stored in the plants. But glycogen is a polysaccharide of, this is the polymer of glucose molecules, which is stored in the animal and human cells. Okay. So cellulose, what was the cellulose? Who knows?
The cell wall of plants.
Of course, this is a cell wall. So it forms the plant cell wall. They provide the rigidity. They provide the turgority. Turgority means the filled with water, okay? So the key difference between the storage and the structure. Storage, glycogen and the glycogen and the starch, they do the energy storage. For example, the glycogen is the energy storage for the animals and the starch is the energy storage for the plants. For the cellulose, it's a little bit different. Cellulose, this is a structural support. This is strong due to the different glucose linkages, okay? And polysaccharide, when we call them polysaccharides, when three or more monosaccharides link together, we call them polysaccharides. Okay, for example, when three and more glucose molecules are linked together, we'll call them as a polysaccharides. Okay, clear?
Yeah.
Any questions to me regarding this? Here you can see this is just one monosaccharide, and this is the polysaccharide. A lot of monosaccharides join it together to form the polysaccharide. And the glucose and galactose, when you join them together, this is the disaccharide, right? Disaccharide, when glucose and galactose join it together, they form the lactose. When glucose and glucose join it together, they form the maltose. When glucose and fructose join it together, they form the sucrose, saccharose. Okay, so let's do them. So first Jahangir, then Munisa, then Gilkhaya, then Mahbub. Okay, so first start Jahangir.
Storage serves as a means of storing glucose in plant in a I think plants. Maybe I'm mistaken.
Yes, exactly. So plants, they store the energy, they store the glucose molecules as starch, right? So plants, but for the animals, they use the glycogen. Glycogen. Yes. Second one, Munisa.
A molecule of maltose is formed from two molecules of uh, glucose.
Glucose. Yes. Third one, Gulhaya. Mahbuba. So glucose and fructose, both of which are monosaccharides, yes, monosaccharides, combine to form a molecule of sucrose, yes, sucrose, which is a sucrose is a
Disaccharide.
disaccharide, yes. Good. Okay, fifth one, Jahangir.
Cellulose is
Cellulose is monosaccharide, disaccharide, or polysaccharide, which one?
I think it's a monosaccharide.
No, monosaccharide is just glucose, galactose, and fructose. Cellulose is,
Polysaccharides.
of course, cellulose is a polysaccharide. It's polysaccharide. Okay, sixth one, Munisa.
Glycogen serves as a means for storing glucose in animals.
Glycogen in animals. Good. Seventh one, Gulkhaya. Mahbuba. Mm-hmm. Yes. Eighth one, Jahangir.
The chemical formula for glucose is
Sucrose is.
a sucrose. Isn't it the same as fructose?
No, sucrose is disaccharide. This is combination of two monosaccharides. Sucrose combination of glucose plus fructose. Okay, this is combination of these two ones. Multiply into two. What we will get?
Um, it's the one that you asked before.
Yes.
I think it was C12 H uh H11O5 H22.
So I said twice as much, right? You multiply it into two and you remove the water. If I
Oh,
multiply it
so
into two, you will
I
get this one, right? You will get
mhm.
this one. But when you remove the water, you remove the 2 here, minus 2, you will get 22. And you remove minus 1, you will get 11. So 12,
uh
22 is coming here and 11 is coming here.
yeah, now I get it.
Okay, good. Nice. So ninth one, Munisa.
Cellulose and glycogen differ in the way that glucose molecules are bonded together.
Yes, glucose. 10th one, Mahbuba. Chemical formula for the sucrose and maltose. They are disaccharides. They are the same. They are identical. Yes, they are identical. They all are like this. Okay. Any questions, guys? We finished the carbohydrates. We finished the proteins. Any questions?
I get no questions.
Okay, if you don't have questions, thank you very much. Our time is over. We can say goodbye. See you next class on Friday. Have a good week. Thank you very much.
Thank
Thank
you.
you.
Goodbye.
See you. Bye.